What if we could peer into the earliest protective bubble of life?
That bubble is the amnion, the membrane that forms a fluid-filled sac around an embryo. Normally, human embryos can't be studied beyond 14 days, so we never see how the amnion develops. But a new study in cells shows we can model it in the lab. Researchers created three-dimensional amnioid sacs from human stem cells. They call them Post-Gastrulation Amnioids, or PGAs. This breakthrough lets us study development from 2 to 4 weeks after fertilization, well past the 14-day limit. It's a big deal because it gives scientists a window into later embryonic stages without using actual embryos. First, a little background. The amnion is a protective membrane that balloons into the sac around the embryo. It cushions the baby against shocks and allows nutrients and gases to pass through before the placenta forms. It was long thought to be just a bubble barrier, but it turns out it can also signal to the embryo.
Why is it so hard to study the amnion in humans?
Because of ethical and technical limits. Human embryos can't be grown in the lab past 14 days, so we can't watch the amnion form naturally. And most stem cell embryo models don't go past implantation or gastrulation. Gastrulation around week 3 is when the embryo cells organize into the 3 layers that become tissues and organs. After that point post-gastrulation, we really know very little about the embryo or extra embryonic tissues. This is where the new PGA model comes in.
Okay, so how did they make these lab amniotic sacs?
The team took human embryonic stem cells and used a 2-step chemical protocol to guide their fate. Over 48 hours, they exposed the cells to 2 signals. After that, the cells self-organized without further tinkering. By day 10, a fluid-filled sac had formed in over 90% of experiments. Remarkably, these sacs grew and expanded over the next 90 days. They even produced and contained fluid that mimics real human amniotic fluid, all without additional signals.
So the cells just naturally formed a little amniotic sac?
Yes, essentially the stem cells arranged themselves into the layers of the amnion. In later tests, the PGAs grew quite large, visible spheres on the order of millimeters, and they remained healthy for months. This 2-step protocol makes the process reliable and reproducible, which is why it's such a powerful new tool.
If you're into cutting-edge biology, hit subscribe.
What did the scientists actually see inside these lab sacs? First, each PGA formed a 2-layer membrane, just like a real amnion. There was an inner layer of amniotic endoderm cells and an outer layer of extraembryonic mesoderm. Microscopy shows it as a translucent 2-membrane sac with fluid in the middle. Inside some sacs, they even observed small yolk sac-like structures forming transiently, which you wouldn't necessarily expect.
Does it really behave like a natural amnion?
Yes, on multiple levels. The fluid composition and nutrients inside the PGA matched human amniotic fluid. The cell types and gene activity in the model closely mirrored natural amnion tissue. In other words, the model mimicked the functional traits of the amniotic sac. In fact, the authors found the gene expression profiles in the PGA endoderm were very similar to that of real human amnion. So structurally and molecularly, these PGAs behave like real extra-embryonic membranes.
What's next for this research?
The team is excited to mix PGA cells with embryo-like cells to study how they interact. Basically, they want to see how an embryo and its amnion talk to each other. This could reveal signals that, for example, tell the embryo what to become after gastrulation. There are also practical applications. The amniotic membrane has valuable medical uses. For example, donated amnion is used to help heal eye injuries, uterine injuries, burns, and ulcers. Right now it depends on donor tissue from C-sections. If PGAs can provide a lab-grown supply of identical membrane tissue, that could be a game changer. In the future, we might grow personalized amniotic membrane on demand. Plus, better models mean better prenatal diagnostics. Scientists could use PGAs to study causes of complications in early pregnancy, like amniotic band syndrome or placental disorders. The possibilities in regenerative medicine and disease research are vast. So, in summary, scientists have created the first lab-grown model of the human amniotic sac after gastrulation. These PGAs will help us peek behind the curtain of early development without needing real embryos. It's an exciting step forward for developmental biology and medicine. Don't forget to subscribe for more science videos and hit the notification bell. Let us know in the comments. What do you think about creating stem cell models of early embryos? Are you excited or concerned? Should we grow more organs in the lab? Finally, suggest a topic. And which other first days of human development would you like us to explore next?